In this video, we're going to talk about the nitrogen cycle and the flow of nitrogen through an ecosystem, in particular from atmospheric nitrogen gas, which we know is very inert and chemically unreactive, through to inorganic nitrogen, which involves ions such as ammonium ions and nitrate ions in the soil, through to organic nitrogen compounds, whereby we have molecules like nucleic acids, DNA and RNA, or proteins made of amino acids that we know contain nitrogen and are found within living organisms. So there's an interchange between the different forms of nitrogen and the nitrogen gas that we find in the atmosphere. So this picture shows us the nitrogen cycle that we need to learn, and we're going to incorporate a lot of these ideas into some of the exam questions. So we're going to start with number 1, top left. Now we know in the atmosphere there's quite a lot of nitrogen. It's about 78.1% nitrogen gas. Now obviously when as mammals we breathe in, we take in 78% nitrogen, but we can't just absorb this nitrogen into our bloodstream and deliver it to our cells because it's not usable. So the air that we exhale and we expire will contain the same 78% nitrogen. And that's because if we actually look at the nitrogen molecule, so N2, we can actually see 3 covalent bonds link the 2 nitrogen atoms. So this triple covalent bond makes N2 gas very stable and therefore chemically inert. So we can't use N2 gas and incorporate that into things like DNA and RNA. Now the next step is number 2 and number 3. So there are 2 types of bacteria. Azotobacter on the left hand side here actually is a free living bacterium that lives in the soil, and Rhizobium on the right hand side here, that is a bacterium that actually lives inside little nodules on the roots of legume plants. And the bacteria, they divide by binary fission and they live within these little root nodules attached to the root system of the legume plants. Now legume plants include alfalfa, clover is another good example of a legume plant, and pea plants as well, or all legume plants. So these will have a symbiotic relationship with Rhizobium bacteria. Now inside these bacteria, Azotobacter and Rhizobium, is an enzyme called nitrogenase. Now this is quite an interesting enzyme, so nitrogenase, because this can take the N2 gas and it can reduce the N2 gas into ammonia or ammonium ions. So the N2 gas becomes fixed into another molecule called ammonia or ammonium ions. Now in Azotobacter, these bacteria can use the ammonia to make amino acids, which they can then use to make proteins. Now Rhizobium can do the same, but they also deliver some of the ammonia to the plants to which they have a symbiotic relationship. So the plants obtain ammonia from the Rhizobium bacteria, and because it's symbiotic, the Rhizobium bacteria obtain sugars, in particular sucrose, that's been made by photosynthesis by the legume plant. So we know ammonia will be delivered to the plants that's been fixed by Rhizobium bacteria in the root nodules. We know the plant supplies sucrose and also some other amino acids to the Rhizobium bacteria. So it's symbiotic. So at this point, the nitrogenase enzyme inside these different types of bacteria has catalyzed the enzyme reaction to fix N2 gas as NH3 ammonia or ammonium ions. Now some of this ammonia, if it's Azotobacter in particular, gets released into the soil. So it starts to make the soil somewhat more fertile. Now the next step in the cycle is, well, we know the plants, they can incorporate the nitrogen from ammonia, if we look at the plant here for a second in the middle, into what we call organic nitrogen compounds. So the nitrogen from the ammonia gets utilized and assimilated into organic nitrogen compounds like DNA and RNA. So we know they're nucleic acids that contain nitrogen. Also, nitrogen is needed to make amino acids. So we could say amino acids, the amine group, and we know amino acids are used to make proteins. We also know chlorophyll contains nitrogen, so we could say chlorophyll, as well as another nucleotide called ATP. So all these organic molecules contain nitrogen that originates, in this case, from the ammonia that has been made by the rhizobium. Now we know herbivores come along and they, like the cow here, and they consume vegetation, the plant material, and they'll take the organic nitrogen from within the cells of the plant material and they'll break down the plant material and start to absorb a lot of these molecules into their blood. These molecules get delivered to the cells of the herbivore, and the nitrogen will get incorporated into new proteins made by the cells of the herbivore, for example. And that's called assimilation, where organic nitrogen is consumed or ingested and then used, the nitrogen is used and assimilated into other organic nitrogen molecules inside the cow, for example. Now, any living organism at some point will die. So we know plants die, and we know animals also die. And when any living organism does die, it will start to be decomposed by what we call saprophytes. Also, any egested fecal material from the herbivore or any type of animal will also be decomposed, and that contains lots of fiber, for example, like cellulose, that can be broken down by the decomposers. Excreted materials like urea, for example, can also be decomposed, as well as all the cells and the tissue of these plants and animals. Now we know saprophytes include both fungi and lots of different types of bacteria. And what these microorganisms do is they secrete, so we can put here secrete enzymes outside these cells, and they digest the dead organic matter externally. And when this organic matter has been broken down, they then can absorb some of the breakdown products into the cells, for example, of the fungus or the bacterium, and they can start to utilize these breakdown molecules. Now, one of the products of decomposition by the saprophytes is ammonia or ammonium ions. So we call this process of decomposition to release ammonium ions from organic nitrogen ammonification. So any dead organic material, any excreted or egested material can be decomposed. The saprophytes have external extracellular digestion. They break down the organic nitrogen molecules like DNA and RNA and proteins. They release the nitrogen as ammonium ions into the soil. Now, some plants can absorb ammonium ions through their root hair cells into the root cortex. But most plants preferentially absorb what are called nitrate ions. So in the soil, there's another process called nitrification. And that is whereby the ammonium ions are converted to nitrate ions, which then makes the soil very fertile. The nitrate ions are absorbed through the root hair cells of all the different plants. So nitrification actually takes place in a two-step process involving two different types of bacteria. Now the ammonium ions in the soil, they first get converted to what we call nitrite ions. So NO2- is a nitrite ion, not nitrate but nitrite. And the bacterium that is responsible for this process is called Nitrosomonas. Now Nitrosomonas is an obligate aerobe, and that means it requires oxygen. So these require conditions in the soil whereby there's lots of oxygen in between the soil particles, because they respire aerobically with the use of oxygen. So Nitrosomonas bacteria, they take in the ammonium ions, they then oxidize the ammonium ions to nitrite ions. These get secreted into the soil, some of them. The second type of bacterium is called Nitrobacter. Now Nitrobacter is also an obligate aerobe. So that requires oxygen because it can perform aerobic respiration. And that takes the nitrite ions, so it absorbs them into the bacterial cell, and it oxidizes these to nitrate ions. So Nitrobacter can utilize the nitrate ions, but some of them will get released into the soil. Now these nitrate ions, so here nitrate ions, are NO3-, and we know the root hair cells of virtually all plants can absorb nitrate ions either by facilitated diffusion or normally by active transport into the root hair cell. So the plants, when they absorb the nitrate ions, they utilize the nitrogen, and they will use that to make more organic nitrogen molecules like DNA, RNA, amino acids, etc. So when there's a high level of nitrate ions in the soil becomes very fertile, because we know the plants can absorb this nitrogen and make this organic nitrogen molecules. So this step here in green with a dotted green line, so we can just kind of look at this here. This is called nitrification. And this is where two types of bacteria are responsible for taking in essentially ammonium ions and converting the ammonium ions to nitrite, which is Nitrosomonas, and then Nitrobacter converts the nitrite ions to nitrate. Now one good way of remembering this is Nitrobacter has a B for bacter. Now if you think A and B, so A would be Nitrosomonas, and B, because Nitrobacter's got a B in it, is the second step, Nitrobacter, nitrites to nitrates. Now nitrate ions in the soil make the soil very fertile, like we said. However, if the soil becomes waterlogged, for example, so there's lots of water within the soil, the oxygen available becomes much less to these microorganisms, so the soil becomes anaerobic when it's waterlogged. Now there is another type of bacterium called Pseudomonas. Now Pseudomonas are obligate anaerobes, so they can actually grow and divide by binary fission when there's no oxygen present, i.e. in waterlogged soils. And what they do is they absorb the nitrate ions from the soils and they convert the nitrates back to N2 gas. And this gets released by these Pseudomonas bacteria back into the atmosphere, and we start the cycle over again. So this last step will occur predominantly when the soil is waterlogged. There's anaerobic conditions with a lack of oxygen. They're obligate anaerobes, and it's called denitrification, and that returns the nitrogen from the nitrate ions back into the atmosphere as N2 gas. And again, we're going to have the cycle repeat itself. So on the previous slide, there was quite a lot of information, and we can actually summarize a lot of that information in 4 key processes. Now, the first key process is the utilization of N2 gas or nitrogen gas from the atmosphere, We know it's chemically inert and it doesn't react with much. So we've got these two specialized type of bacteria that can fix N2 gas as ammonia or ammonium ions. So we could say ammonia, or we can actually add in NH4 plus ammonium ions. So this is a reduction reaction. N2 gas gets reduced to ammonia or ammonium ions, and we know the enzyme inside these bacteria is called nitrogenase. So the nitrogenase enzyme catalyzes this reaction. Now this nitrogenase enzyme is actually very sensitive to oxygen. So if there's too much oxygen present, it can actually inhibit the enzyme. So these two bacteria have mechanisms to prevent the oxygen levels inside the cells getting too high and inhibiting the enzyme. Now Azotobacter, for example, have a very high rate of aerobic respiration, and we know from the respiration topic that oxygen is required and is utilized and used up in aerobic respiration is the final electron acceptor. So the high rate of aerobic respiration inside Azotobacter prevents oxygen from building up inside these bacterial cells, and so it doesn't tend to have its nitrogenase inhibited. Now Rhizobium, that we'll see a bit later, we know this is these bacteria are inside the root nodules on the root system of legume plants. Well, inside the root nodules, they've actually got quite a pinkish appearance, and that's because the legume plant actually makes and synthesizes hemoglobin. So normally we hear about hemoglobin in the blood of mammals, and it carries oxygen around the circulatory system. Well, some legume plants can also synthesize hemoglobin proteins, and these get delivered to the Rhizobia bacteria, and we know hemoglobin binds the oxygen and it prevents the oxygen from building up around these bacteria, so it keeps it relatively low, and again it prevents nitrogenase from being inhibited. So we call this first step nitrogen fixation, and the end product is ammonia or ammonium ions. And we know the ammonia can also be utilized by the legume plant, which makes it a symbiotic relationship because the Rhizobia bacteria receive sugars and other amino acids, etc. from the plants. Now the second step is called ammonification. So if we just label these, so nitrogen fixation was number 1, ammonification is now number 2. Now we know once organisms have nitrogen and they can incorporate and assimilate the nitrogen into organic nitrogen like DNA and RNA and proteins, if they are to die, that organic nitrogen gets decomposed. So we know saprophytic fungus or fungi and saprophytic bacteria secrete enzymes to digest the organic material outside the organisms, outside these cells. The organic nitrogen is broken down enzymatically, so the DNA, the RNA, the proteins, the amino acids, the ATP will be hydrolyzed and broken down, and the organic nitrogen gets released into the soil as ammonium ions. So any dead animals or plants, when they're decomposed, we know the bacteria and the fungi break them down and we get ammonium ions into the soil. The third step is nitrification. Now on the previous slide we did say there were two types of bacteria, so Nitrosomonas and Nitrobacter. Now Nitrosomonas we know takes in the ammonium ions released into the soil as a consequence of ammonification, sometimes called putrefaction, decomposition, and Nitrosomonas converts the ammonium ions into nitrite ions here. So NO2 minus is nitrite ions. Now these nitrite ions, if they're released into the soil from Nitrosomonas, they're absorbed by Nitrobacter. So we know B is the second step, if you like, A and B. B for Bacter, Nitrobacter. They take in the nitrite ions and convert them to nitrate ions. Some of these nitrate ions get secreted into the soil, so it makes the soil nice and fertile. Because we know plants like to absorb nitrate ions through their root hair cells, and we know the plant, it will utilize the nitrogen from the nitrate ion here, and it will incorporate that nitrogen into organic nitrogen, so the plant can then start to make its own DNA and RNA and amino acids and proteins and chlorophyll, for example. Now we know the fourth step is if we were to get waterlogged soil, it creates more anaerobic conditions with a lack of oxygen. We know the nitrifying bacteria like Nitrosomonas and Nitrobacter, they require oxygen, so these can't continue to grow in the absence of oxygen. But Pseudomonas, this denitrifying bacteria at the bottom of the page, that can survive in a lack of oxygen in what we call waterlogged soil. So waterlogged soil, so a lack of oxygen. They're obligate anaerobes, that means they don't need oxygen, they can respire anaerobically. They absorb the nitrate ions from the nice fertile soil. They convert the nitrate ions into nitrogen gas. It's the nitrogen gas that gets released back into the atmosphere. So if we go all the way up to step number 1, we're going to restart the cycle again. So focus on this first nitrogen fixation step in a little bit more detail. Now we know the two bacteria, Azotobacter, that's free living in the soil, and Rhizobia, they live in the root nodules of the legume plants. And we know they take in the nitrogen gas, N2, and they fix this into ammonia, or we could say ammonium ions. It actually depends on the pH inside the bacterial cells. And we know some of the ammonia, if it's from Rhizobium, is used by the legume plant to make organic nitrogen. So this is the root of the legume plant. It might be alfalfa or clover or pea plant. And we can see here the root nodules. So there's quite a few of them in this picture. Now the root nodules are actually part of the plant itself. So if we took a cross section, say here, X to X, we would see the typical cross section of a root. But if we took a cross section maybe here, a little bit further up, we're going to take a cross section of the root nodule as well. So this is what that would look like in cross section. Now we know the root has an epidermis, which is a single layer of cells on the outside here in green, and some of those are specialized as root hair cells, and they allow the uptake of mineral ions such as magnesium, potassium, and also nitrate ions, some ammonium ions, and water. And we know inside the epidermis in the cross section of the root is the cortex, and this can have anywhere from 50 to several hundred cortex cells depending on the size of the root. And in the middle of the root we get the stele. So this central structure made of several different tissues is called the stele, and in blue here we've got the endodermis, and we know the endodermis cells are specialized because their cell wall has suberin injected into them, which makes the Casparian strip. Now we know in the middle of the stele, this tissue, it looks like a star shape or an X shape. These cells that make up this tissue constitute the xylem tissue, and they're xylem vessels and xylem tracheids, as well as parenchyma or parenchyma we could say, and fibers. Now in orange, we'll do it in purple. In purple, this is the phloem tissue, and that's made of sieve tubes, made of sieve tube elements, and also companion cells. So we know the xylem transports water and dissolved minerals up the plants from the roots up the stem, and we know the phloem, the sieve tubes, transport or translocate sucrose and amino acids up and down the plant. So we can see in this cross section that some of the cortex cells have actually been infected with rhizobia bacteria, and this is the internal part of the root nodule. Now we can see extending from the stele, so this bit here in the root, is vascular tissue, and this will be made of phloem tissue and xylem tissue. So it allows sugars that have been made in the leaves of the legume plant to be delivered down the phloem, down into the root, and then into the root nodule. So sugars and some amino acids get delivered to the bacteria inside the root nodule itself. It also allows the ammonia or ammonium ions to be delivered from the bacteria into the root of the plant, up the roots, and to the rest of the plant. Now ammonia and ammonium ions are actually dissolved in the xylem tissue, or the xylem vessels of the roots, so they get transported up the plant to where they're required and where they're needed to make organic nitrogen. Now, if we look at this picture on the left-hand side, we can actually see a photograph of the root nodules themselves. So these are little houses for rhizobia bacteria. Now, the root nodule, as we said earlier, it is part of the plant itself, and the rhizobia infect the cortex cells inside the root nodule. Now, if we were to chop open one of these root nodules, what we would see is this kind of reddish, pinky colour here in the middle of the root nodule. Now, we said earlier that the nitrogenase enzyme inside the rhizobia bacteria, it is inhibited somewhat by higher levels of oxygen. So the normal process of nitrogen fixation is nitrogen gas plus protons and electrons, because we're going to add hydrogen to the N2 gas. It's a reduction reaction. That does require some ATP, so we're going to put ATP in there. So it's an energy demanding enzyme reaction, and what's produced at the end of this is ammonia. So we're going to put ammonia here. So you don't need to know this reaction, it's just to help understand what's happening. And also, if we were to add in the 16 ADP and the 16 inorganic phosphate, because we know the phosphate group, the terminal phosphate group from the ATP, is hydrolyzed, and that releases a small packet of energy that drives this enzyme reaction. So we could put here nitrogenase. There you go. So we know it's an enzyme reaction, and we know we get ammonia at the end of it. Now, because this enzyme is inhibited somewhat by oxygen, the plant itself does make hemoglobin. Now, we know hemoglobin does have an affinity with oxygen, a quite high affinity. So it does bind a lot of the oxygen that surrounds the rhizobia bacteria in the cortex cells of the root nodule. And this carries away some of the oxygen, so it prevents the buildup of oxygen inside the rhizobia bacteria, and therefore it prevents too much inhibition of nitrogenase, and we continue to get nitrogen fixation. So it's one mechanism as part of the symbiotic relationship between rhizobia and the legume plant itself to allow the rhizobia to fix nitrogen optimally. And it's the hemoglobin that gives the red colour, as it would do to mammalian blood as well. Now, you don't need to know how the root nodules are actually formed on the root system of the legume plants, but I will talk through it just to give you a little background information of how that occurs. So the plant cells, we know on the outside of the root are a single layer of epidermis cells, and some of these epidermis cells are specialized to form or what are called root hair cells, and each root hair cell has a long extension called the root hair. Now, these cells can release what are called flavonoids. So these are just chemicals released by the root hair cells. Now, the rhizobia bacteria originally are actually outside the roots in the soil. So these little blue dots might be the rhizobia bacteria. Now, the flavonoids released by the plants, they stimulate the rhizobia to secrete chemicals actually called nod factors. Now, these chemicals actually bind to receptor proteins in the cell membrane of the root hair cell. So imagine this was a root hair cell with its long root hair extension. Now, the chemicals that are secreted by the bacteria bind to proteins in the cell membrane of the root hair cell, and that causes calcium ions to actually enter the root hair cell. Now, we know if calcium ions enter the cytoplasm of a cell, it's going to lower the water potential of that cell. So what happens is water enters the root hair cell by osmosis, and that causes the root hair cell to become turgid, and it actually causes it to kind of curl around, like we can see in this picture here. So the root hair cell has become turgid because of the entry of water by osmosis, and it's curled around, and that actually circles around the rhizobia bacteria. So these little blue dots that you can see in the picture. Now, the rhizobia, they inject more proteins or different types of chemicals that causes the plant roots to develop what are called infection threads. So these infection threads go down the length of the root hair cell, and they extend into the cortex of the roots of the legume plant. Now, the rhizobia bacteria can move down these infection threads, and once they get into the cortex, they infect the cortex cells of the root of the plant. So we know the layer of cells from the epidermis on the outside through to the stele is called the cortex. So these cortex cells, they become infected with rhizobia. And inside the cortex cells, these bacteria can divide, and they produce more chemicals, causing the cortex cells themselves to divide. So infection with rhizobia causes the cortex cells to divide. And this starts to form the nodule. So we can see here, this is a nodule starting to form where the cortex cells continue to divide. We know inside the cortex cells we've got bacteria called rhizobia. Now, the nodules, we know, and we can see here, they do start to become pink because the cortex cells start to produce what's called hemoglobin, or it's a certain type of hemoglobin that binds a lot of the oxygen to keep it relatively low, so it prevents inhibition of the nitrogenase enzyme inside the rhizobia bacteria. Now, over time, the nodule continues to develop, so it does get bigger, so it becomes visible by eye if we look at the roots. And it also starts to develop vascular tissue. And we know that vascular tissue contains xylem vessels, xylem tracheids, and also phloem tissue. So that is going to allow the movement of ammonia down the xylem and into the xylem in the actual middle of the root. So ammonia and ammonium ions is transported up the xylem through the root structure to the rest of the plant. We also know through the phloem tissue, sucrose is delivered to the bacteria because they need to respire. So the bacteria can actually utilize the sugars delivered to them, which includes sucrose and therefore glucose and other hexose sugars. And they can use that for respiration because bacteria need to respire to release energy in the form of ATP. So we've talked about nitrogen fixation, and we know a little bit about the nitrogenase enzyme, and in particular rhizobia and the root nodules of legume plants. But I'm just going to focus on, if we go over to the right hand side, so we can see here nitrate ions on the right hand side. And we know soil is very fertile if it's got lots of nitrate ions in it. And that's because nitrate ions are absorbed by the roots of all plants, so legume plants as well as non-legume plants. And we know the nitrogen from the nitrates is going to be used by the plants and assimilated into organic nitrogen. So we know these plants have to make their own DNA and RNA and amino acids and therefore proteins, etc. And they need this nitrogen to be able to do that, which predominantly does come from nitrate ions. So we're going to have a bit of synoptic knowledge, and we're going to go over how nitrate ions are absorbed through the root hair cells into the xylem in the root. So on the left hand side here, this picture, it shows the root of a non-legume plant, so a regular plant. Now we can see these projections here are the root hairs, and they're projections from root hair cells. So some of the epidermis cells are specialized as root hair cells, and these predominantly allow the absorption of mineral ions, like we said earlier, NO3- nitrate ions, or even maybe sodium or calcium, etc., and also water by osmosis. So if we did a cross section of the root that we covered in year 1, we can see the layer of epidermis cells is this outer layer here. So I'll just highlight that in purple. So it's a single layer of cells on the outside of the root. Some of these cells, like this one here, are specialized, and these are the root hair cells. So any of these epidermis cells with a big long extension called the root hair are actually the root hair cells. There, you can go there. Okay, so if we actually look at a root hair cell in a little bit more detail, what we can see is this picture down at the bottom. The protoplast, or the living contents of the cell, are made up of the cytoplasm and the vacuole. So we can see this bit here is the cytoplasm of the root hair cell. We can see the vacuole here. Now the vacuole is a storage structure, and it does store mineral ions, salts, sugars, some amino acids, etc. And it contains what's called sap. So we could put sap there, vacuole sap. Now this membrane here is called the tonoplast, and that's the phospholipid bilayer of the vacuole. And we can see this line here is the phospholipid bilayer that actually forms the cell membrane of this root hair cell. On the very outside, we've got the cellulose cell wall. So this thick layer is the cellulose cell wall on the outside of the root hair cell. Now, the vacuole does actually extend down the inside of the root hair, as does the cytoplasm. So we can see here, this is the cytoplasm, thin layer of cytoplasm, and inside it is the vacuole. The cell wall does extend down the root hair as well. So this elongation is the root hair, but it's a bit thinner compared to the cell wall in the main body of the root hair cell. Now, how does nitrates or nitrate ions get into the root hair cell? Well, we know in the cell membrane there are carrier proteins. So if I put these in purple in little dots here, these would be carrier proteins for nitrate ions. And these carrier proteins in the cell membrane of the root hair cell, they pump in nitrate ions from the soil water into the cytoplasm or the vacuole of the root hair. Now, because normally the concentration is higher in the soil water compared to inside the protoplast of the root hair, we know this is going to be active transport, and it is going to require the energy released from hydrolysis of ATP. So this is an active pumping of nitrate ions through a carrier protein into the vacuole and cytoplasm of the root hair. Now, once these mineral ions and the nitrates build up in the root hair, we know that's going to reduce or make more negative the water potential inside the root hair protoplast, vacuole, cytoplasm. Now we know water is going to move from high to more negative water potential by osmosis. So as long as mineral ions like nitrates are going to be pumped into the root hair, water is also going to enter the root hair cell by osmosis down what we call a water potential gradient. Okay, So when we talk about the movement of water by osmosis from high to more negative water potential down a water potential gradient, we don't refer to water concentration. That's nothing to do with osmosis. It's literally from the values, the water potential values, from higher to more negative. And water actually, because it is polar, it predominantly goes through channel proteins in the cell membrane of the root hair called aquaporins. Okay. Now, once water enters the root hair cell, that's actually going to make the water potential less negative because water is going into the protoplast of the root hair cell. So the water potential of the root hair cell actually goes slightly above the water potential of the adjacent cortex cells here. So these are the first cortex cells. So because the water potential is slightly higher in the root hair cell compared to the adjacent cortex cells here, water goes down its water potential gradient into the more negative water potential cortex cells. Now, as water enters these cortex cells, the water potential goes above the more negative water potential in the next row of cortex cells. So again, water is going to move down a water potential gradient into the adjacent cortex cells. And as water moves into these cortex cells, again, it increases the water potential above the more negative water potential in the next cortex cells. So again, the water moves by osmosis down a water potential gradient into the next cortex cells. And this continues all the way across the cortex, which are these cells here, until the water and the dissolved minerals and the nitrates, etc., get to the endodermis. So this layer of cells here, this single layer of cells, is called the endodermis. Now these cells are specialized because in the cell wall they have the Casparian strip made of suberin. So we know water can't move through the suberin in the cellulose cell wall. And we know mineral ions can't also move through it. So the apoplast pathway, which is the movement of water and dissolved minerals, nitrates, etc., through the cell walls of the cortex cells, that is now prevented because of the Casparian strip. Now, there is a role and a reason for this. So the mineral ions, the only way they can get from the endodermis cell wall into the cytoplasm of the endodermis cell is through the cell membrane. And because they are charged, they have to go through carrier proteins to get through the cell membrane into the endodermis cell. So the plant can regulate how many carrier proteins and what type of carrier proteins are expressed in the cell membrane of the endodermis cell. So if the plant requires some more magnesium, it will increase expression of magnesium carrier proteins in the cell membrane of the endodermis cells. Now, we know as the mineral ions get pumped into the endodermis cell, it makes it more negative in terms of water potential. Water now follows by osmosis into the endodermis cell, and eventually we know mineral ions are pumped into the xylem, and so water follows down a water potential gradient. So these structures here that you can see are actually called xylem vessels, and we know xylem vessels have lignified walls. So these are big long tubes made of dead xylem cells with lignin injected into the cell walls. Now lignin is essentially waterproof, so water can't just escape once it's got into the xylem vessels, and it gets pulled up through transpirational pull. On the inside of the xylem vessels there is actually a hydrophilic lining. So lignin is predominantly hydrophobic, but part of the molecule is actually charged, which faces on the inside of the xylem vessel, and that allows adhesion of the water molecules to the inside of the xylem vessel. And we know water is sticky. It can hydrogen bond with each other. So water hydrogen bonds through cohesion, which allows the water to be pulled up in a continuous stream up the xylem vessels. Now these cells here that make this tissue, these are actually the sieve tubes and their companion cells. So here we would get sugars being translocated down into the root, and the root we know is a sink because those sugars will get used in respiration to allow things like cell division, etc. So that's a basic recap of the root structure of the different plants. Now, 3 pathways. So we've mentioned the apoplast pathway, which is probably about 95% of water and mineral ions go through this apoplast pathway. It's the quickest of the pathways, and it goes through, we can see here in red, the cellulose cell walls of the cortex cells until it gets to the endodermis cell. Now the Casparian strip, you can see here as these black blocks made of suberin, and that restricts or prevents the apoplast pathway. So we know the mineral ions have to get into the endodermis cell through the cell membrane, and that occurs, so I can do the cell membrane here in green. There you go. It's the cell membrane. Through, as we said, carrier proteins, and water follows into the cytoplasm of the endodermis cell by osmosis from the cell wall. Now the other mechanism, or pathway if you like, for water and minerals to progress through the cortex is through the cytoplasm and the thin strips of cytoplasm that link adjacent cortex cells together. So we've got the root hair cell on the left. Water and minerals enters the cytoplasm. It goes down through the plasmodesmata into the adjacent cortex cell. It gets drawn across the cytoplasm down a water potential gradient. It goes through the plasmodesmata to the next cortex cell, through the cytoplasm, and it continues to do this pathway all the way through the plasmodesmata, through the endodermis cell, through the pericycle cell, and eventually, because the minerals are pumped into the xylem vessel to create a negative water potential, water follows the minerals into the xylem vessel and up the xylem tissue. Now, one other pathway we could mention is when mineral ions and water get into the cytoplasm, sometimes they move into the vacuoles. So this water can actually move through the cell wall, through the cytoplasm, and into the vacuoles, because we know these are large storage structures inside the plant cells, so they form a large part of the plant cell. And this water and the dissolved minerals can continue to move from plant cell to plant cell across the vacuoles, and eventually the water and minerals will end up in the xylem vessel, and that is called the vacuolar. So vacuolar pathway. So we know nitrogen is absorbed into the roots of plants, predominantly as nitrate ions, some of it as ammonium ions. But what is the nitrogen actually used for in the plant then? Well, we know it's needed to make organic nitrogen compounds, and the first example we might want to give is amino acids. So we know this inorganic nitrogen is used to make organic nitrogen molecules, and we know amino acids, we can actually draw out their structure because we've done that in year 1 in core concepts. So we can draw the amine group, NH2, we can draw the central carbon atom, we know that that's connected to a variable group, we've got a hydrogen atom down the bottom, and then we've got our carboxyl group on the end here. So that's a typical amino acid. We know there's 20 different types of amino acids in nature, so we could say 20 amino acids, and they all differ due to the nature of their variable group. So there's 20 different variable groups that we find in the different amino acids. Now we know amino acids are subunits for proteins to be made, and we know proteins are made through a process called translation in the ribosomes or at the ribosomes of all plant cells. And we know proteins include different types of molecule like hormones, like enzymes, etc. Now another example is nucleic acids. So we know nucleic acids involve or include the DNA and the RNA. We know DNA is found inside the nucleus of all plant cells, and it is actually arranged as chromatin because it's wrapped around histone proteins, and that's the genetic material, the DNA. We know there's several different types of RNA, so we're going to go ribosomal RNA, and we know that's needed to help make the ribosomes, at which translation occurs. We've got transfer RNA, which transfers in specific amino acids to the ribosome for the process of translation. So we know tRNA has an anticodon that's complementary to the codon in messenger RNA. So messenger RNA is the molecule that is transcribed. So we could say mRNA we know is transcribed inside the nucleus from the template strand of a particular gene. It moves out of the nucleus through a nuclear pore and it attaches to a ribosome in the cytoplasm of the plant cell, or maybe attached to the rough endoplasmic reticulum if that protein is to ultimately be secreted from the cell or to form part of the cell membrane. So there's three different types of RNA, and we know DNA and RNA are both nucleic acids. We know each subunit is made from three parts. So it has a phosphate group, it has a pentose sugar, and we know in DNA it's deoxyribose, and RNA it's ribose. And we also know the third part of the nucleotide is the nitrogenous base. So nitrogenous bases in DNA can be one of four types. The nucleotide might have adenine as its nitrogenous base, it might have thymine, it could have guanine, or it could have cytosine. Now we know RNA does not contain any thymine, but instead it will have uracil, which has a slightly different structure compared to thymine. So nucleotides we know need nitrogen to be able to be made, and that nitrogen is needed for the nitrogenous base. Nucleotides are the subunits or building blocks of nucleic acids. Now we know ATP is actually a type of nucleotide, but it does differ to DNA and RNA nucleotides in that it has three phosphate groups. So ATP, the T stands for triose phosphate, so three phosphate groups. The pentose sugar in ATP we know is ribose, very similar to RNA, and the nitrogenous base this time is adenine, so A for adenine, ATP. ATP actually is called adenosine triphosphate, so we can put in here adenosine triphosphate. Now we also know the plant needs to make photosynthetic pigments, and these include chlorophyll. So when we do the photosynthesis topic, we'll see that there's two types of chlorophyll, chlorophyll A and chlorophyll B. Chlorophyll A is the primary photosynthetic pigment, and chlorophyll B is an accessory pigment. There's also other pigments called carotenoids, which include beta-carotene and xanthophyll. But as long as we know nitrogen is needed to make chlorophyll, which absorbs certain wavelengths of light. Now this is a different diagram. It's exactly the same information. This is the one in the class booklet. So we can look at the different processes here, maybe highlight some of them now that we know a bit more about them. So we might want to start with nitrogen N2 gas in the atmosphere that we've said. We know it gets fixed by rhizobia bacteria in the root nodules of legume plants like alfalfa and clover. We know Azotobacter can also fix nitrogen, and that's a free living bacterium in the soil, so that releases ammonium ions into the soil. We know rhizobia, they do release some of the ammonia, and that gets used by the legume plant to make organic nitrogen, like we just mentioned. We know plants do get eaten by animals, so the organic nitrogen is consumed by these animals, and the organic nitrogen is then used to build organic nitrogen molecules inside the animal itself, so DNA, RNA, amino acids and proteins. We know all plants and all animals die. So dead organic material will be decomposed by saprophytes. So we can say saprophytes here, which are the fungi and the bacteria, and they have extracellular digestion and they absorb the products of their digestion. We know animals ingest material, including a bit of cellulose and fiber, things they can't break down and digest themselves. They also excrete certain molecules like urea, which can also be decomposed. Now, we know decomposition can also be called ammonification because the organic nitrogen is released into the soil as ammonium ions. We know most plants absorb nitrate ions through their roots, so the ammonium ions are converted by nitrous ammonius to nitrite ions, so NO2 minus, and that's an oxidation reaction. So these are obligate aerobes because they require oxygen for aerobic respiration. The nitrite ions in the soil are converted to nitrate ions, so we can go NO3 minus by nitrobacter. We know nitrobacter because they've got a B in it. It's the second reaction. So nitrous ammonius A, nitrobacter, B for bacter, B is the second reaction. They take in the nitrites and we get nitrates released back into the soil, some of them. We know the nitrate ions are taken in by the roots of all plants and used to make organic nitrogen. We know if the soil is waterlogged, the nitrate ions are taken in by denitrifying bacteria called Pseudomonas. They convert the nitrate back into nitrogen gas, and that goes back into the atmosphere. So it goes back into the atmosphere as nitrogen gas, and the cycle obviously repeats itself. So we can start to look at exam questions now using the knowledge about the nitrogen cycle. Now this question gives us quite a simplified picture of the nitrogen cycle, and the first question A says name process P and Q. So if we look at P, we're going to start with NO3 minus nitrate ions, and these are being converted into N2 gas. So we know nitrate ions are needed to make the soil fertile, and they're actually being produced by nitrification. But the reduction in nitrates to N2 gas is, as we know, a process of denitrification by Pseudomonas. So the conversion of nitrates in the soil, which actually makes the soil nice and fertile, to back to nitrogen gas, we know is denitrification. It occurs in waterlogged soils by anaerobic bacteria called Pseudomonas. Now Q, on the other side, if we start with N2 gas, Q is the conversion of the N2 gas into ammonia or ammonium ions. So this must be nitrogen fixation, where the N2 gas, which we know is non-reactive, it's very inert, it's being fixed with the addition of hydrogen inside a Zotobacter or Rhizobia and converted to ammonia. Now the next part of the question says each year a total of 3 billion tons of ammonia is converted to nitrate. So this step, if we do this maybe in green here at the bottom, it says 3 times 10 to the power 9, which is 3 billion tons of ammonia, is converted to nitrates. Now we know this step here, we can start to annotate the picture a bit. It's called nitrification, and that involves nitrous ammonius and nitrobacter. So nitrous ammonius takes in ammonia, converts it to nitrite. Nitrobacter takes in nitrite and converts it to nitrate ions. Now the next part of the question says only 2 times 10 to the power 8 tons of ammonia is produced from nitrogen gas. So we can put that up here. 2 times 10 to the power 8 tons of ammonia is being made through nitrogen fixation by Azotobacter and Rhizobia. So there's a discrepancy between the amount of ammonia being produced, which is 2 times 10 to the power 8, and the amount of nitrates being produced, which is a lot higher at 3 times 10 to the power 9, so 3 billion tons. So where does the extra ammonia come from? which is then converted to nitrates. And it says explain the difference in these figures. So there must be another process occurring here to make ammonia and release it into the soil for it to be converted into nitrates. Well, we know when plants and animals die, the dead tissue, all the organic nitrogen like DNA, RNA and proteins, is broken down through ammonification. So decomposition of that dead organic material releases ammonium ions or ammonia into the soil by the action of saprophytes, fungi and bacteria. So they decompose the dead plants and dead animals. The organic nitrogen is released into the soil as ammonia, and that's where the extra ammonia comes from, which is then converted to nitrates through nitrification. So ammonia, let's have a look at the answer, is formed by the decomposition or the ammonification of dead plants and animals through saprophytes, otherwise known as saprobionts. It's exactly the same, through decomposition. And the molecules that get broken down, as we said, are organic nitrogen compounds, or it might be nitrogenous waste like urea or other molecules that contain nitrogen. Right, the conversion of ammonia, question C, to nitrate involves oxidation. So we know nitrification is the conversion of ammonia to nitrites and nitrites to nitrates. What evidence in the diagram supports this statement? So why is NH3 oxidized to NO3-? Well, we could say two things. We could say oxidation is loss of hydrogen. So we know NH3 loses its hydrogen, which is oxidation, because it also loses electrons. Or we could say it's the addition of oxygen to the nitrogen. So we get oxygen being added to the nitrogen atom to make the nitrate ion. So we could say either of those two things. So oxygen is added, or we could say hydrogen is removed, and that's therefore an oxidation reaction. Now another question then. Much of Indonesia is covered with forest, and large areas of the forest have been cleared and planted with oil palm trees to be used in the production of fuel. So quite often companies will plant trees, and after a certain period of time they'll cut the trees down and use the wood as a form of fuel, or the wood might be used to make things like ethanol, which is a biofuel. Question A. In these forests, nitrogen in dead leaves is made available to growing plants by the action of bacteria. Describe the role of the bacteria in making the nitrogen in dead leaves available to growing plants. So dead leaves then, we know they're going to be decomposed. We know the bacteria and the fungi that do this are called saprophytes. We know they've got an extracellular mode of nutrition. We know they're going to break down the organic nitrogen in this dead plant tissue, and that's going to be converted to inorganic ammonia or ammonium ions. So let's have a look at the mark scheme then. We mentioned saprophytes, and we said they break down the organic nitrogen compounds like DNA. So we're going to give some specific examples of the organic nitrogen molecules, proteins. We said it's extracellular digestion because these saprophytes, fungi and bacteria secrete enzymes from the cell into the soil. They digest these molecules outside of the actual organism. We said ammonia or ammonium ions is produced from the organic nitrogen molecules. Right, we now know ammonia or ammonium ions is converted to nitrites first and then to nitrates through the two types of bacteria, Nitrosomonas and Nitrobacter. So we're also going to give these two in the answer as an example. And this is called nitrification. And we could have mentioned the type of reaction. Well, we know it's an oxidation reaction. We know NH3 or ammonia is converted to NO2- ions, and then the nitrate ions, which are absorbed by the roots of all plants. And we know that's an oxidation reaction. So there's the full mark scheme for that answer, and it's a 5-mark question. Okay, so this slide shows a previous exam question. It's actually an essay question, and it said describe, so we're going to use examples where we can, how the nitrogen cycle allows nitrogen in one plant to become available to another plant. And then it says, describe the various roles of nitrogen in plant metabolism. So there's two main parts to this essay question that we could really think about. And the first one is up to this point here. So how does the nitrogen cycle allow nitrogen in one plant to become available to another plant? Well, we know the nitrogen in one plant is in the form of what we call organic nitrogen, like we've mentioned. So we can start to mention these types of molecules like nucleic acids, nucleotides that are used to make the nucleic acids, and ATP is a nucleotide, amino acids and proteins and chlorophyll, etc. It's organic nitrogen. So how is that made available to another plant? Well, we know plants predominantly take up nitrogen as nitrate ions, NO3-. So we need to start to think about what happens to a plant, the organic nitrogen, and how is that made available to another plant in the form of nitrate ions. The second part of the essay, we're going to talk about the roles of nitrogen in plant metabolism. Now, metabolism just really means all the biochemical reactions that occur inside plant cells. So it might be the production of certain molecules like amino acids, like proteins, like chlorophyll, like DNA, RNA, etc. Anything that contains nitrogen. So the first part of the essay then, let's have a look at the answer. Well, we know plants, they shed their leaves, and these leaves will die and be decomposed. We know plants also die, so the whole of the plant at some point will also be decomposed. Now we know consumers feed on plant material. So herbivores, for example, feed off plant material, vegetation, and we know herbivores and any other animal that comes along and eats the herbivore will also die. So the nitrogen in the plant itself and the nitrogen inside consumers that feed off plant material will also be decomposed. So we can then start to talk about the decomposition of this dead material, and that involves, like we mentioned earlier, fungi and bacteria, which are saprophytes. And we can again talk about extracellular digestion. We can start to talk about some of the organic nitrogen molecules that get hydrolyzed by these digestive enzymes. So proteins hydrolyzed to their subunits, amino acids. We could even mention deamination of amino acids, because we know this type of reaction is needed to remove the nitrogen from the amino acid. So if we drew in an amino acid here, it's the amine group, variable group, and the carboxyl group, like that. So in order to remove the amine group, that is called deamination, and that will start to then help to form ammonia or ammonium ions, and this is called ammonification. Then we talk about nitrification. So that's where the ammonia or ammonium ions in the soil is converted to nitrite, and then nitrite to nitrate. We know the two types of bacteria, so Nitrosomonas and then Nitrobacter. We know they're obligate aerobes because they require oxygen for aerobic respiration. And we know plants absorb the nitrate ions from the soil. So we've talked all about that through active transport. This is then transported through the xylem tissue up to the cells of the plant, and the nitrogen is used to make organic nitrogen. Now the second part of the essay, we could talk about the amine group, the NH2 group in amino acids. So we know nitrogen is needed for that. We know amino acids are subunits of proteins, which are essentially polypeptide chains. So amino acids are linked by peptide bonds between the carbon of one and the nitrogen of another amino acid. That's the peptide bond. We know nitrogen is part of nitrogenous organic bases that form parts of nucleotides. So we know the base is a part of a nucleotide. We know ATP is also a type of nucleotide, but with 3 phosphate groups. We know it's part of chlorophyll because that's an organic photosynthetic pigment that has nitrogen in it. And actually we could add as well, when you do the respiration topic and photosynthesis topic, the coenzyme NADP, which is a hydrogen carrier needed in photosynthesis, that is actually, it does contain nitrogen just as ATP does. Now we know some farmers use fertilizers and they apply these to their crops, and that allows the soil to become more fertile and therefore the crops to grow to a greater extent. So what are fertilizers? Well, it might be dead plant material that's been decomposed by decomposer organisms, like this example in the top. It could be things like manure that's been left to decompose, and that might form compost as well as the plant vegetation. We can have fertilizers where chemicals have been added, and that is applied to fields normally as a spray. But most fertilizers will contain certain key elements. Now, nitrate ions we know are needed for making organic nitrogen molecules. So we know the nitrogen here is utilized by the plant cells to make amino acids and therefore proteins, nucleic acids like DNA and RNA, and things like chlorophyll. Now, phosphate ions, PO4 3-, they're very important in nucleic acid synthesis because we know DNA is double-stranded. It has 2 sugar phosphate backbones. So the phosphate ions are needed to make the sugar phosphate backbone of each strand of DNA. And it's the same for single-stranded RNA. The sugar phosphate backbone requires phosphate. Now we know phosphate ions are also needed to make phospholipids. So a phospholipid is made of glycerol attached to 2 fatty acids by ester bonds and attached to a single phosphate group. And it's the phosphate group and the glycerol that make the polar head of the phospholipid. Now we know all membranes are made from 2 layers of phospholipids called the phospholipid bilayer, and that goes to make cell membranes as well as membranes inside the cell that form organelles. So things like the rough ER membrane, the smooth ER membrane, the nuclear envelopes, and we know nuclear envelope is 2 membranes, as well as things like the thylakoid membranes that we find inside chloroplasts. Now phosphate groups are also needed to make ATP. And we know ATP has 3 phosphate groups. The 2 terminal phosphate groups, phosphoanhydride bonds, they're high energy bonds that release a small packet of energy when they become hydrolyzed. Now we know potassium ions are very important in stomatal opening in normally the lower epidermis of the leaves of these crop plants. Now we know potassium ions are pumped into the 2 guard cells during the day by active transport. This lowers the water potential inside the guard cells, so water moves in by osmosis. It makes the guard cells turgid, and because the thicker inner portion of the cellulose cell wall is thicker than the outside portion, it forces the stomatal pore open during the day. Now sulfur is an element that quite often is added to fertilizer, and it's a component of certain amino acids such as cysteine. So cysteine is an amino acid that has sulfur in its variable group. So the end of the variable group has this SH group, and that's going to be important in forming disulfide bonds within certain proteins to give them a 3D shape, maybe a tertiary shape in their protein, or even a quaternary shape. So cysteine is an example of an amino acid with sulfur in its variable group, and it's important to make disulfide bonds. So this is a nice example of an AO3 question. Now we know AO3 is about analysing data that we're given either as a table, so we might see some data in a table, or as in this case in a graphical format, and then we need to draw conclusions about that data and what it tells us about the experiment that's been conducted. So AO3 is really about drawing conclusions from the data that we're given. So in this experiment, what's happened is the scientist has looked at the growth of a certain species of crop plant by looking at the dry mass, or the increase in dry mass over a certain length of time under different treatment conditions. So there's 5 different treatment conditions on the bottom. There's a control, So every experiment we're going to have a control in there. We have fertilizer with just nitrogen added. We have fertilizer with nitrogen and phosphorus added. We've then got another treatment with nitrogen, phosphorus and potassium. And lastly, we've got a fertilizer with nitrogen, phosphorus, potassium and sulfur added. There you go, sulfur. So we've got several different treatments. Now, the way that you'd set up this experiment, you would actually use test tubes. So you'd have a minimum of 3 test tubes per treatment. Probably use some more because then we can still generate a mean and we've got more data points for each treatment. Now, in each test tube, you would place a solution, and that solution would contain mineral ions, maybe some growth factors, obviously water, as well as seedlings of the same size and the same species. So we're going to place our seedlings of the same size and the same species into the solution. Now, obviously, we're then going to add our treatment. So the first treatment we might want to do is add a fertilizer only with nitrogen. So the concentration of the fertilizer with the nitrogen is going to be the same for each of the 3 tubes. We're also going to have other control variables. So we would need to keep the light intensity the same, because obviously the seedlings do photosynthesize and make glucose, and glucose is then respired by the plant cells to release energy, and that allows cell division and therefore growth. So we're going to keep the light intensity the same. We're also going to keep the temperature of the solution the same and the ambient temperature surrounding the seedlings consistent. We're also going to use a certain concentration of carbon dioxide and keep that consistent because that's needed. The carbon from it's needed to make glucose in photosynthesis in the Calvin cycle. We're also going to keep the pH of the solution the same for each of the solutions that we're going to use. So the only thing that we're going to change is the nature of the treatment, so the addition of fertilizer. And there's 4 different fertilizers. One will just have nitrogen, one will have nitrogen and phosphorus, one will have nitrogen, phosphorus and potassium, and the last one will have nitrogen, phosphorus, potassium and sulfur added to it. Now, obviously, over a certain period of time, it might be 1 week, we are then going to remove the seedlings. We're going to place them in an oven and dry them off to evaporate all the water content from the seedlings. And then we're going to measure the dry mass of the seedlings. And that allows us to plot the data on this graph here. So the y-axis going up the left-hand side, that's always the dependent variable. Okay, so the dependent variable that we measure. And in this case, it's the dry mass of the seedlings after a certain length of time. Now the x-axis along the bottom is always our independent variable. And we normally have 5 independent variables that we test, but it can vary. In this case, we've got a control, and then we've got our 4 different treatments. So what we're going to use for the control, question 1. Well, we always should really have a control in the experiment. And in this case, we're just going to add water. So we're not going to add, so there's going to be no fertilizer here. It's just going to be water added to the solution instead of any of the elements. And then we're going to measure dry mass over maybe a week. The other treatments, we're just going to add fertilizer with nitrogen, then fertilizer with nitrogen and phosphorus, etc. Okay. So how do we analyze and make conclusions from this graph? Now, it's very easy to say the best fertilizer to use is the fertilizer with nitrogen, phosphorus, potassium and sulfur. So this one at the end here. However, that's not enough detail. We need to start looking at the data in a bit more detail. And the way we do that is to look at the dry mass of the controlled seedlings where no fertilizer has been added, and then compare these values with the other 4 values that we have. So if we look at the different values, so you're obviously going to take a ruler and read off the y-axis for each treatment, and that's going to give you a value of dry mass in grams after a week. So we can start to compare these different values. So we've said the control just with water, with no fertilizer, gives us 11 grams after a week. Now, treatment with just fertilizer with nitrogen added gives us 45 grams. So we want to compare this value, these two values together. So we actually now can see that between the control and between the treatment with just nitrogen, we actually get a 34 gram increase in dry mass. Now, if we then look at the next treatment with N and P, so if we just say the control treatment on the left, N is treatment 1, NP is treatment 2, NPK is treatment 3, and NPKS is treatment 4. So if we look at treatment number 2, that gives us 50 grams of dry mass after a week. So by adding N and P, we know it does produce a higher dry mass compared to just N on its own. However, the effect of phosphorus we can work out because we already know the effect of nitrogen from treatment number 1, 45 grams. Now, if we add N and P, we get 50 grams. So actually, just adding phosphorus gives us a 6 gram increase. Sorry, 5 gram increase, isn't it? 5 gram, yeah. So the effect of phosphorus here gives a slightly increased growth from 45 grams up to 50 grams. And that's quite a small value in comparison to the effect of nitrogen, which actually gives us a plus 34 gram increase. And then we can do the same for treatment number 3. So if we look at N, P, and K here, we know the dry mass we're going to get with N and P, because that's treatment number 2. So when we treat with N, P, and K, treatment number 3, we can actually work out the effect of potassium on growth. So we go from 50 grams with N and P up to 52 grams with N, P, and K. So the effect of potassium, if we draw that in, gives us a plus 2 gram increase because we've already treated the seedlings with N and P, number 2. So N, P, and K, so we've added potassium this time, gives us a plus 2 gram increase. And we can also work out the effect of sulfur on plant growth. So treatment number 3 is N, P, and K. Treatment number 4 is N, P, and K plus sulfur. And that gives us from 52 grams up to 54 grams. So again, that gives us a plus 2 gram increase in dry mass just through the addition of sulfur, because we're comparing column number 3, N, P, and K, with column number 4, N, P, and K plus sulfur. Now, what we want to do in the exam answer is to express these as percentage increase. So percentage increase. So this is always an exam question that does tend to crop up. And what we need to say is the new value minus the initial value divided by the initial value. So we're now going to compare the addition of just nitrogen on its own to the control. So this value here versus the control value of 11. So the new value is 45 grams when we add fertilizer with nitrogen minus the control value with no nitrogen, which was 11 grams, divided by 11 grams, because that's the original value, times 100, because we want a percentage. So we'll put percent there. So if we just do the maths, we're going to get 309% increase in dry mass just through the addition of nitrogen alone. Now, if we do the same calculation for the addition of N and P, okay, we actually want to look at the effect of just the phosphorus. So if we compare the treatment number 2, that value of 50 grams, with treatment number 1, 45 grams, we can then work out the effect of just phosphorus on its own. So again, we could do the same sort of calculation. The new value is 50 grams for N and P minus 45 grams that we had just for N divided by 45 grams times 100%. And that will give us a value of, if we just work that out quickly, 11% increase. So the effect of just phosphorus on its own gives us an 11% increase in dry mass. Now, the next one, we're going to do N plus N, P, and K. And here, we want to look at the effect of potassium. So we're going to compare treatment number 3 with treatment number 2, because we know treatment number 3's got potassium in it and treatment number 2 hasn't. So that's going to give us 52 grams is the new value, minus 50 grams, because that's N and P, treatment number 2, divided by 50 grams times 100. And if we just do the maths there, again, we are going to see, so it's 2 grams divided by 50 times 100 is 2% increase. So the effect of potassium is a 2% increase in dry mass. And again, it's actually going to be the same, or very similar, isn't it, for the last one. So if we do N, so plus N, P, K, and S, that allows us to work out the effect of sulfur. So here the maths is going to be 54 minus 52, because 52 grams is NP and K, 54 grams is NP and K plus S, divided by 52 times 100, and that's going to give us 3.8%. So we can start to see the effect of these different elements on plant growth. We know by far nitrogen gives us the biggest increase in dry mass, that big 34 grams, which is 309%. The addition of phosphorus, potassium, and sulfur does give us small increases individually. And we know if we were going to choose a fertilizer, obviously we would choose NPK and S, because that gives us the biggest overall growth. So one of the problems with the use of fertilizers by farmers on their cropland is that if they're applied at the wrong time of the year, or too much is used throughout the year, some of the nitrate ions in the soil water, if there's excess rainfall, they can be washed away from that soil into surrounding waterways like ponds and lakes. And we call this leaching. So excess nitrate ions in farm soil can be washed away into local waterways, and that can result in a thing called eutrophication. Now, if you ever see a lake or a pond or a stream with this kind of algal bloom, so this green color on the surface, the health of that water is actually quite poor, because these are algae that grow on the surface of the water. And we're going to see why in the next slide. So we can start to talk about the process of eutrophication. Now, we know farmers do use fertilizers with lots of nitrates in them. If there's heavy rainfall, sometimes these nitrates that are dissolved in the soil water can leach into surrounding waterways like lakes and ponds. The nitrate concentration in these waterways then increases. Now, the nitrates can be taken in by algae, and we know algae are going to use the nitrogen in the nitrates to make organic nitrogen molecules like DNA and RNA and proteins, etc. And that allows the algae to divide and replicate, and so we get what's called an algal bloom on the surface of the lake. So this green layer here, now that blocks sunlight penetrating the water. Now at the bottom of the lake we've got aquatic plants. Now we know light, because water is transparent, the light can penetrate the water normally, and so these plants can photosynthesize, but when there's an algal bloom it blocks the light, so the plants do not receive adequate sunlight, they can't photosynthesize. if they can't photosynthesize, they can't make glucose, which obviously they need for respiration and energy, but they actually also need glucose to make other organic molecules like amino acids and lipids. So these plants tend to die off. Now, as the algae increase on the lake, surface of the lake, obviously there's going to be competition. So some of these algae also die off because they can't get all the nutrients they need. So we've got decomposing bacteria in the water, and we know these are saprophytes, so extracellular digestion, and they break down the dead algae and also the dead plant material saprophytically by secreting enzymes into the water. Now, these are aerobic bacteria, so that means they require oxygen in order to respire aerobically, to be able to divide by binary fission. So these bacteria require oxygen for growth and division, and they respire aerobically. And obviously, if they take in a lot of the oxygen from the water that's been dissolved in the water, the oxygen content of the water does drop dramatically. Now, any aquatic animals that live within the water, like these fish, if there's not sufficient oxygen in the water, they can't get sufficient oxygen across their gills, which is their gas exchange surface, into their blood, and therefore eventually they're also going to die. So the health of the pond or the lake becomes very poor due to the lack of oxygen because of these bacteria that decompose the dead algae and the dead plant material. Now, obviously, through respiration, carbon dioxide is released into the water, and that kind of effect on the pH. So the more CO2 we've got, the lower the pH becomes. Now, as the oxygen gets used up, that's going to allow anaerobic bacteria to start to thrive because they do not require oxygen for respiration, and that's called eutrophication. So we can represent eutrophication as a flow chart. So fertilizers, we know, do leach into water courses like ponds and lakes, etc., from farmland, especially if there's heavy rainfall. That's quite a key word to use, the leaching. And we know that fertilizers include nitrate ions because the nitrogen is needed by plants. It's absorbed, the nitrates are absorbed through the roots of plants, and the nitrogen is used to make organic nitrogen. We know if the nitrate content of the water increases, there's quite often an algal bloom. So these algae divide rapidly because they can use the nitrogen from the nitrates. That forms a surface on the water that blocks sunlight, so the aquatic plants towards the bottom of the lake don't receive sufficient light. So the amount of light is not enough, and therefore they don't photosynthesize to the same extent, and they tend to die off because they can't make enough glucose. Decomposer bacteria and fungi then decompose the dead organic matter, including dead plants and also dead algae. We know the bacteria respire aerobically. That means they take in oxygen from the water for aerobic respiration to make glucose, because any living organism has to make has to respire glucose to release energy, and that creates what's called a biological oxygen demand within the water course. Aquatic animals that would normally take in the oxygen from the water, maybe fish through their gills, then die off because of that lack of oxygen. And due to the lack of oxygen, any anaerobic bacteria can then start to flourish because they do not require oxygen for respiration. They respire anaerobically. So how can farmers overcome high nitrate levels in waterways and therefore help to prevent eutrophication? Well, they can actually restrict the amount of fertilizer that they add to the soil in the first place. So if there's less fertilizer being added, then there's actually less nitrates in the soil that could be leached into the water course. They might only want to apply the fertilizer when the crops are actively growing, so maybe a certain time in the year where the crops grow best. So crops grow best. So they're kind of limiting the amount of fertilizer they use because they're only going to apply it during the active period of growth. And that really does ensure that the nitrates in the fertilizer are being taken up by the crops when they're going to need it the most. It's also possible to dig drainage ditches for these minerals to concentrate in. that surround the cropland. So that prevents the minerals, maybe the nitrate ions, from leaching into the watercourse itself, and they tend to concentrate and accumulate within these ditches that can then be emptied. That can, however, have sometimes a knock-on effect on the invertebrate biodiversity, because obviously you build in these drainage ditches, which does disrupt some of the land further.